Alright, so we'll go through the ear pretty quickly here. Again, a reminder that the anatomy is just going to be in the lab, whereas in lecture we'll talk about the function, the physiology. So note here there's the external ear up until the tympanic membrane, and there's going to be the middle ear here that includes the ossicles, which are the three tiny bones in the ear, and then inner ear is this part here in green with your cochlea that looks like a shell, and the semicircular canals, and then here the eustachian tube. So sound is coming from sound waves that are basically compression and rarefaction of air molecules. So they get compressed and then they get spaced out, and that is what the sound is that we're able to interpret. So sound has the pitch and the intensity. So the pitch is determined based on the frequency of the vibration. So here in this chart you can see a low note has a low frequency versus a high note has a really quick frequency. And then intensity is just the amplitude of the wave. So a really soft tone is a pretty small wave versus a very loud tone is a very big wave. Okay, so amplitude. So we measure sound intensity with the decibel. You guys are probably familiar with this. It's a logarithmic scale, so it's not exactly equal. So 30 decibels is 10 times louder than 20 decibels. Some examples down here, just kind of FYI. 10 decibels, rustling of leaves, versus a loud concert is 120 decibels. Okay. So this outer part of the ear here is called the auricle or the pinna, and this is what kind of gathers those sound waves and sends them into your external auditory meatus, the ear canal. The middle and inner ear, we can see the bones here, and then here is the startings of the cochlea. So in the middle ear, it's basically air. There are the little bones in here. Oops, don't look up bones. Little bones in here, the malleus, incus, and stapes. And basically what they do is they vibrate. So sound comes in and it hits your tympanic membrane, which is your eardrum, which vibrates. The malleus, which is attached to the tympanic membrane, vibrates the incus, and that vibrates the stapes, which is attached to something called the olive oval window here. So basically, the middle ear is taking vibrations from the outside and bringing them in. This is also where most ear infections are found. We call this otitis media. So the little ear bones are called ossicles. There's the malleus, incus, and stapes. You guys will need to know these in the lab. And then something interesting here, the tympanic reflex. So your tympanic membrane, like we just said, vibrates in response to, it vibrates because of the sound waves. And there are little tiny muscles that attach to the bones here. And if there's a really loud, quick sound, those muscles will flex and they'll keep the vibrations to a minimum so that the bones in the ear don't break. So a little protection here from very quick, like a gunshot or something like that, very loud, quick sounds. So here you'll see the eustachian tube or auditory tube. Either term is fine. This is how we connect our ear to the nasopharynx, which is in your throat. So when you're on a plane or something going up the mountains and your ears pop, that's because this tube is allowing the pressure to equalize from inside and outside of your ear. So equalizing air pressure here. So this is just a little drawing here looking at the middle ear. So like I said before, the vibrations come from the sound waves. Here's the tympanic membrane, and it causes the ossicles here to vibrate, and they press against something called the oval window, and then that sends vibrations into the inner ear here. So these vibrations can be very, very fast. So here in the inner ear we have the cochlea and the vestibular apparatus. And within the cochlea is the organ of Corti, which is the organ for hearing. So this picture is a cross-section of the cochlea here. And we can see there are 3 different chambers that each have fluid in them. So if we look a little bit closer here, this is the organ of Corti. So there's the basilar membrane that's attached to a bunch of little hair cells. And the hair cells are attached to the cochlear nerve that goes to your brain. So what happens is the fluid will come and it will rush against these little hairs and cause them to bend. When they bend, they send signals. It causes depolarization, and they send signals to the brain of what just happened. So different hair cells in different parts of the ear allow us to hear different tones. So the organ of Corti here is basically the organ of hearing. There are 2 membranes, the tectorial membrane and the basilar membrane. The tectorial membrane is on top here. It's rigid, it's not moving. Basilar membrane is flexible and it's moving from the sound vibration. Little hair cells come up here from the basilar membrane and then attach to the tectorial membrane. So basically, the stereocilia are bathed in this fluid called endolymph, and when the vibrations come in and cause movement, the stereocilia bend. When they bend, it opens ion channels. Potassium flows in, which depolarizes the cell, and that releases neurotransmitter. So you can see here in an unstimulated stereocilia hair, the gates are closed, the mechanically gated channels for potassium. So if you remember, mechanically gated channels open and close in response to like physical stress. So here they're closed, and then when the hair cells bend, the gates open, allowing that potassium to rush in, causing depolarization, and then the release of neurotransmitter, which will send a signal to the brain so that you know what you just heard. So determining the pitch. So depending on where the hair cells are being moved, that will tell us where the pitch is. So the cochlea is shaped like a shell, and as you wrap around and get into the innermost part of the cochlea, that's the part that's going to sense the really high pitches, versus the outermost regions are sensing the lower pitches. So it's determined based on the location of the hair cells being moved. So you can see here, shaped like a shell. As we get towards the center, these are higher pitched versus on the outside, lower pitched. The way we determine amplitude is based on the number of hair cells being triggered. So the more hair cells being triggered leads to more potassium rushing in, which leads to more action potentials and more neurotransmitter being released, so the brain can interpret this as louder. I'm going to skip this here. So the innervation of the ear comes from this nerve that you guys know as vestibulocochlear. The vestibular nerve comes off of the vestibule here that we'll go through in a second, and the cochlear nerve comes off of the cochlea. They combine together to form one cranial nerve. You guys can go ahead and look at this link on your own if you so choose. Here's the pathway for controlling hearing. I'm going to pass through this here. So on here, what I'd like you to know is which parts of the brain are helping to control hearing. So your inferior colliculus is responsible for, like, if there's someone slams the door, you would turn your head and look. If someone says your name, you turn and look. It's kind of that response. And then actual perception is in your temporal lobe. So if you're listening to someone sing and you're like, hey, they're not a great singer or whatever, that would be happening in your temporal lobe. So the cochlea is here, the cochlear nerve comes, and it decussates, and it crosses over. So then it goes to the auditory center. So because of this, if there's damage to either auditory center, you don't get unilateral deafness, so deafness on one side, because there's so much crossover. So the vestibular apparatus is the part of your inner ear that helps you with balance. So equilibrium is your control of coordination and balance. This is from your vestibular apparatus. And there's two different types of equilibrium you should know. There's static equilibrium, which is your perception while you're stationary, and the dynamic equilibrium, which is your perception of movement. So the saccule and the utricle are different chambers within the vestibule, and they contain the macula that senses your movement forward, so linear acceleration. In the macula, there are little hair cells, very similar to in the cochlea, and they do the same thing as in the cochlea. So when they bend, they open and they can send signals. Different cells will be triggered, bent, based on what type of movement you're having. So you can see here this gymnast is standing upright. You can see there's very little stimulation here in the macula. So all the hair cells are upright, versus when she turns upside down, you can see the fluid comes down with her and it bends all of these little hair cells, causing those potassium channels to open, rush in, and then causes action potentials in the brain. Semicircular ducts have fluid in them, and they're helping to sense rotation. So you can spin around in circles, and this is the part that's going to sense that rotation. So here the hair cells bend with rotation, and that causes the wall to move at a different speed than the fluid. So it causes that rotation and triggers different cells to bend. So you can see here, as she's turning, this is kind of pushing the fluid one way versus the canal is going the other way. That's causing the action potentials here in the hair cells.